Yeah, hello and welcome back to our PML School. My name is Bernd Wendt and I'm here with Chris Mail and Dan Weiner, and today we have a very popular topic, allometric scaling, and we've seen a huge interest in this session. And this session will be delivered by Dan Weiner. Before we move on, just to let you know that there's a Q&A panel on the right-hand side of the WebEx console. So if you have questions or comments, please post them there, and either we will respond to that during our session, or we have a session at the end where we explicitly respond to questions, and later on we will also post the responses to any remaining questions on the Certara forum as usual, and we also will make the material available, like slide deck and a link to the recorded session. But now I guess it's time for starting the session, the lecture. Dan Weiner, please take over.
All right, thank you, Bernd. So today, as Bernd said, we're going to talk about allometric scaling, but before we actually get into an example showing how to use Phoenix when nonlinear to perform allometric scaling, we thought it might be useful for at least some of the attendees to see a bit of the background with regards to allometry. So we're going to start with a bit of a mini lecture. Much of the materials in the mini lecture is also contained in the textbook that Johan and I co-authored, and then we will go through in detail the example PK29. So why do we want to do scaling? Well, I'm sure everyone's aware of the reasons for that, but generally what we're trying to do is figure out an appropriate dose in man. And before we jump in man, of course, we study the drug in animals first, and then we want to try to scale those results to man. Part of the reason why we want to do this is to also predict an actual concentration time course in man, and whenever possible, come up with some measures of variability that we might expect to see when we actually get into man as well. So what do we use when we do allometric scaling? Well, it depends on the various situations in the data that's available. Ideally, we'd like to be able to scale something like an IC50, and generally we work with data, as we said, that's generated in animals, so preclinical data. So as you know, one of the other ways that we generally do scaling is using in vitro methods where we try to predict the corresponding Vmax and Km in man. So there we're sort of going from test tube to man. But as you'll see in this lecture, it's also possible to do scaling going from in vivo to in vivo, where the first in vivo, of course, are the results that we get in animals, and then, of course, we're going to extrapolate onto man. So what is allometry? So if you looked it up and just looked at the definition, it's really defined as the relationship between body size, and you could use various measures of body size, but body weight is the one that's most commonly used, and then some physiological variables, such as clearance. But as we'll see, there are many parameters that often we are able to scale, and we'll go through some of those. So to help motivate why scaling can actually be useful, here's a very well-known, I guess, plot showing absolute and relative oxygen consumption, and it's one of the slides that's often used to illustrate the value of doing allometric scaling. So if you look at the axis on the right, there we're looking at absolute oxygen consumption in liters per hour, and it corresponds to the black dots, and you can see it's plotted against body weight. And if you just looked at this slide and only looked at absolute consumption, it really looks like as animals get heavier and larger in size, and you can see humans are right around this area here, they correspondingly use up a lot more oxygen. But on the other hand, if you were to scale these data and look at oxygen consumption, but on a liter per hour per kilogram basis, and that's the red line, there you're actually able to see that the heavier animals actually more efficiently utilize oxygen than do lighter animals. So scaling can help provide clarity to some of these types of issues. Well, what can scale? Well, first off, and I've got a note to remind us at the bottom, that when you do allometry itself is based on empirical data and not biological rules, and you have to remember that. And at the slide at the end of the deck, we'll talk about situations where you might be able to predict where allometric scaling may or may not work. But when it does work, you're often able to scale things like volumes relative to body weight, just raised to the power of 1. In other words, there's a 1-to-1 correspondence. As body weight increases, you see a corresponding increase in volumes. Metabolic capacity, such as oxygen consumption we just looked at, or clearance, blood flows, turnover rates, and the like, that scales according to body weight to the 0.75 power. Measures of time generally scale according to body weight to the 0.25 power. And frequencies and rate constants often scale body weight to the minus 0.25 power. Now, you can scale both drug and system parameters. So you could have a drug parameter like IC50, and you can scale that. Of course, if protein binding differs across species, you'll need to adjust for that. But in many instances, you can also scale system parameters such as Kn and Kout as well. So here we're looking, for example, at a turnover model where the drug is acting to either stimulate or inhibit the production of response. And for these types of models, for many drugs, Kn does scale according to body weight to the 0.75. The pool size, denoted as ASS, scales according to body weight to the power of 1. And Kout scales, because that's a Kout parameter, it scales according to body weight to the minus 0.25. And here, for example, you can see here's the same turnover model we're looking at before, where we're inhibiting production of effect. And the results that you see in rat may differ markedly from man, and that's because when you scale across species, we have to adjust for body weight to the various powers as indicated on the bottom of the slide. So now we're getting into some basic allometric equations. So here we have, some PK parameter or other parameters such as turnover, Kn or Kout, and generally we'll model it as indicated here with Y, would be as modeled as some intercept times body weight to a power. And then you can see if you take the logarithms of both sides, you can linearize this. So here, for example, we're looking at clearance across 4 species. And if you were to plot body weight versus clearance, that intercept here, and we're looking at low clearance, I'm sorry, not the intercept, but the slope then would be B here for clearance. Generally in notation, we denote the intercept and slope for clearance as A and B, and for volume, we denote as C and D. Now, if you know, for example, that you need to target the same AUC across species. So for example, maybe you know what an effective AUC in rat is and you want to get the same exposure in man. If we use the allometric equations and set the 2 areas equal and just do a bit of algebra, what we see then is that the dose that you want to use in man would be the corresponding dose in rat, and then you look at the ratio of body rates raised to the B power, where B again is the scaling exponent for clearance. Now, when you are doing allometry and doing scaling across species, the range that you're able to collect data on for body weight is extremely important. So, for example, if we had only obtained data in monkeys, dog, and man, and we looked at the slope here, we're looking at, say, something like lifespan versus body weight, the slope might look as indicated by this red line. On the other hand, if we had data on other species that went further out to the left, in other words, lighter animals, there, if you look at the full slope, you see it's markedly different than what it might look like if we just looked at a slope over a much smaller species. So it is important when you're doing scaling to try to get as wide a body range. Choose animals to scale that have as wide a body range as you're able to do. And the other thing that's sort of an artifact of when you do regression modeling, the prediction errors are much smaller around the center of the data than they are when you extrapolate. So that's something you have to remember. Man is usually out on the end of our extrapolations, and you can see these confidence bands do get wider. So it does show that we are getting larger prediction errors associated with man than we would for some other species that's closer to the center of the actual data. So here's a one-compartment bolus IV model, and then we've just substituted volume and clearance for the allometric scaling values. So for concentration as a function of time, we have the expression as denoted here. And then you could take the integral of this from 0 to infinity and come up then AUC 0 to infinity would be this expression as well. And again, we're going to actually fit some data to this particular model in just a little bit. So there are measures of physiological time that have been developed in the literature. So things like if you're looking at times such as associated with half-life or aging, et cetera. And the way to think about this is that the thought is that small animals spend their lives at a much faster pace than larger animals because generally they live longer periods of time than larger animals do. So you kind of get biased results depending on what you're plotting if you don't adjust for these differences in life expectancy. And one way to adjust for this is to adjust for this via changes in body weight. So Dedrick proposed two ways to plot data. One is a simple Dedrick plot and the other is a complex Dedrick plot. In the simple Dedrick plot, you would, instead of plotting T on your x-axis, you would plot T divided by body weight. One here is the assumed value of the exponent for volume. So you'd use 1 minus B, where B is the corresponding slope or exponent for clearance. And then this is the corresponding adjustment you'd make on the y-axis to do the simple Dedrick plot. When you do the complex Dedrick plot, there you're allowing the exponent of volume to be other than 1. We call that exponent D, such as indicated down here. So you would plot T divided by this adjustment in body weight. And again, on the concentration scale of the y-axis, instead of assuming D is 1, you use whatever that estimated value is. So we're actually going to regenerate these plots today. This is PK29 on a data set. And you can see in this particular instance, we have data on mouse, rat, and man. If you did the simple Dedrick plot, where you assume that the slope for volume is 1, you can see the data don't overlay very well. But if you do the complex Dedrick plot, where D is allowed to float, and you actually numerically estimate the value of D, we see that we get a very nice overlaying of the data. So we'll actually show how to compute those in Phoenix in just a bit. All right, so now we're leading into PK29. In this case, we've got actually 5 species, rat, mouse, monkey, man, and dog. Here's the corresponding doses, and all the doses were micrograms, and the dose was administered as an IV bolus. Here's the corresponding body weights. Now, in this particular instance, for illustration and purposes of the lecture today, we're fitting mean data from these species. But it's becoming much more common these days to take the individual data and actually do a full population analysis of that, where you would use NLME as opposed to Phoenix Wind Non-Len. The actual model's the same. The only difference is you're allowing inter-subject variability on the parameters if you're doing the NLME modeling. So here we see we have just the basic equations for a 2-compartment model. Except that for each of the 4 parameters in this model, we're allowing them to be scaled across species according to their corresponding body weights. So the parameters in this model are actually not clearance, volume, distribution clearance in the secondary volume, but rather the parameters are A, B, C, D, G, and E. So we estimate those parameters as fixed effects, and then for each animal, we can actually predict its corresponding volumes and clearances. So here's plots of the data. I first plotted the raw data, the full data that I have, and as you can see, it's kind of hard to see what's going on from a compartmental perspective. So then I just rescaled the data and only went out to 150 minutes, and now it's very clear that there's a biphasic decline across all the species, and that's That's why then we're going to go with this two-compartment model as indicated here, just based on the plot. So the first thing that I would recommend doing in a situation like this is a non-compartmental analysis. You would sort by species. And here I've actually estimated clearance and VSS for each of the species. And then I went ahead and plotted body weight versus clearance, again, using the NCA clearance. And you can see they're tending to fold very nicely on a straight line. So that suggests for this drug, the allometric scaling is probably going to work. The other thing you can do then is actually do a regression on this line just based on NCA results, where you would fit the log clearance to log of body weight. And then when you do that linear regression, and I will show you how to do this in Phoenix in just a bit, this intercept then is the log of A right here. So it's the log of A. And then this slope is actually B. That's the exponent for clearance. So then we can use these as initial estimates when we do the combined model later. So that's one way to get initial estimates. So I'm going to go through another way as well. So when I have data like this, I don't just, throw all the data into one big pot and try to model it, right out of the gate. Rather, I would fit each species separately first. So here, we're going to use a two-compartment model. We're going to use these as a sort key, okay, and fit each species separately. And if you do that, here's the observed and predicted values overlaid. And then here's the corresponding estimates. If you look here at clearance, and I've got the covariate body weight on here as well, but the estimated clearance in man is 0.49, it's 0.01 in monkeys, and I should have used significant digits here, not decimal places, but then it's somewhat smaller in mouse and rat. But the point is we're able to estimate these separately, and now we can do linear regressions fitting the log of each of the parameter versus the log of body weight, and that will give us initial estimates then for the intercepts and slopes. And that's what I've done here. And again, I'll show you how using the LinMix module in Phoenix how to do these linear regressions. But this is the output from fitting the linear regressions. That's the intercept and slope, and then I used just a data transformation in Phoenix to exponentiate the intercept. So that gives me the actual A, B, the corresponding actual intercepts. So I take the analog of them to get it. But the actual values here that we do in the regression are the actual exponents directly. All right, so these then become the initial estimates in our combined fit. So this is what the combined model looks like. Basically, I would start with a two-compartment model using the GUI interface, and then you can unclick the random effects because here we're just doing a naive pooled analysis because we just have one profile for each species. Then convert it to text. I have to add a covariate statement, and I have to add these statements in because we're not estimating volumes and clearances as fixed effects. We're estimating A, B, C, D, E, G as fixed effects, and then computing volumes and clearances as indicated here. That's our allometric equations. And if you do that fit, here are the final parameter estimates and their CVs, and you can see for this data set, we get a very precise estimates of all the parameters. They're all less than 20% with the exception of C. And C here is the intercept for volume. That's why I just sort of lined them up over here on the left for you as a reminder. And then here's the observed versus predicted values, and then here's the QQ plot for the residuals. So you can see the fit of the data is actually very good. And we're able to estimate, again, these fixed effects with quite good precision. Okay, so maybe what I'll do now is, before I jump into the hands-on session, here's the corresponding Dedrick plots. We looked at those earlier. And I'll just finish with this slide, and then we'll go into hands-on session. But as I mentioned, allometric scaling is an empirical methodology that we know works for many drugs, but it won't work in all instances. It's not a biological law. So how can we identify possible exceptions where the scaling may not work? Well, if you know that you have binding differences across species, that could cause you problems in terms of scaling. There may be cross-species metabolic differences, different genomic effects, different SIFs and metabolism that could cause differences that will lessen the likelihood that they're able to do scaling as a function of body weight. You may have very complex time concentration profiles. You know, they may oscillate or have other non-standard sort of functionalities that you can't normally mimic using standard volume and clearance models. If the systems are nonlinear, you may have difficulty scaling them. If you have low intrinsic clearance, many times those compounds have difficulty with scaling. And just looking at just the behavior of the data using some of these allometric plots can also help identify situations where the scaling may not work. All right, so that's the sort of mini lecture leading into the exercise. So now I'm going to share the Phoenix application. And then, Bernd, could you just quickly confirm that you're able to see the Phoenix screen?
Yeah, it's nice and clear.
All right. So here's what the data file looks like. I've got columns that correspond to time and plasma concentration. I did number the groups, which are the different species. And we saw we have 5 species that are available. And then I've also included body weight in kilograms for each of the species as a covariate. Now, if this had been a population analysis where you have multiple animals per species, you would just have another column, an ID column. All right, so that's the basic data. Well, one thing I always like to do when I am doing modeling across projects, I like to create separate workflows for exploratory data analysis and modeling, and then I may have workflows that correspond to other things that maybe I want to do in particular instances. So in this case, I have 3 workflows, one for the exploratory data analysis, one for the modeling, and then at the end, I used the fitted values for B and D and then did the Dedrick plots to show you how to do those. So in the EDA, it's very straightforward. Again, I just plotted the data, overlaid the species, and then I replotted the data. So I could see, you know, what's really happening at an earlier time point. It's very easy to do that on your x-axis. Instead of doing automatic scaling, I did a custom scale and just told it to go from 0 to 150. So it's very straightforward to do this. And then this confirms the biphasic decline for the species. Then I did a non-compartmental analysis. The thing here is you want to, of course, sort by species, but carry along your body weight. And the reason why you want to carry along the body weight, and again, it's IV bolus, so then when we go to the results, if you scroll your results all the way over, we have a column of body weight. So now if I want to plot, for example, clearance versus body weight, I don't have to do any data merges or anything like that because body weight is already on the same data set as the NCA data. That data just made a nice table out of the results using the table function. And then as we looked at in the slides, I created a plot of clearance based on the NCA results versus body weight. And then I used the data wizard just to compute the logarithm of body weight. We can look at the setup for that. So again, if we look at transformation here, I just did a transformation and just took the logarithm of body weight. Why? Because I need that when I'm doing the linear regressions in order to get initial estimates of the parameters. And now if you send your data to one of the modules that's called linear mixed effects, essentially that's a very powerful lin mix. It's equivalent to like lin mix in SAS. So it does linear mixed effects, although in this case we're not doing mixed effects, but we are doing linear regression. So for the x-axis, I'm taking a logarithm of body weight. So that's my regressor. And then I also told it to take the logarithm of the y variable. Why? Because remember when we did the allometric scaling, it's like a e to the b power. So to linearize that, I have to take the log of a plus b times body weight. So I have to take the logarithm of each parameter. So I can specify those, run this model, and then if we look at results, here's the intercept and slope. So this value here, this is the logarithm of a for clearance, and then this value here is the slope for clearance across species. So what we see based on the NCA clearance is that clearance does scale, but body weight raised to the 0.717 power. Okay, so it's a little less than 1. So these aren't our final estimates. These are just one way to get initial estimates using the LinMix module. I know a lot of our users, because you're primarily fitting PK/PD models, you may not even know the LinMix module is there, but it's very handy, not only for doing these kinds of regressions. It'll do multiple regression if you needed it, but you can also use it to do analysis of variance and bioequivalence assessment as well. All right, so now I go into modeling, and as I indicated on the slides, the first thing I do is to model the data by species. So here I'm using the 2-compartment model, which is what the plot of the data suggested. Due to the range spread of the concentrations, I started out with a multiplicative error model. And then I unclicked population, and then I'm just fitting time versus C odds, and I'm sorting by species. So I unclicked population because we're not fitting them simultaneously. We're using it as a sort key. For the initial estimates, I took these from doing LinMix on all the parameters that we saw previously. Let me go here to parameters, fixed effects. So these were the values that were suggested, and actually they're kind of close to 1. Clearance, I used, looks like I used 0.1, although our point estimate, it was 0.1, it was closer to 0.1 for some species, and then it's higher for other species. So you go ahead and run the model, and when you run the model, so it's actually running now. It'll blink once for each species because we're using the sort key, but it actually fits pretty quickly because, again, we just have one profile for each species. And then you'll get your thetas out for each of the species. And then what I did, I merged these outputs, thetas, with body weights, and then I created and put them into a table. Okay. Now these then will become the inputs that I'll put into the LinMix module. Where before, remember I did LinMix but only using clearance from NCA. Now I'm going to use LinMix using the outputs from the individual fits from across species. So I'm going to fit that regression for each parameter. You see here, I'll look at the data set again. So I'm going to sort by parameter, and then I've got the estimated values of the parameter for each species. I can go ahead and fit that model, and if I do that, then I get all the intercepts and slopes. And now the next thing I want to do is pivot the intercepts and slopes. Let me just rerun this real quick. That'll get rid of the red. So again, I'm going through all this fitting of the individual species just to come up with the initial estimates for the final model. So I'll pivot this. Now that I have the data pivoted, I can send this to the data wizard and just exponentiate the intercepts. So if we look here in the data wizard, see I'm just taking the exponent, exponentiating each of those intercepts from LinMix. And now these values here, these are the intercepts and slopes that we use in the final fit. And then I just made a nice little table out of those as well. Now, to get to this final model, there's a couple ways you could do it. Because here we're just doing naive pooled and I don't have random effects. The way I did it, I sent it to a Phoenix model. And then in the Phoenix model, I turned off closed form because I want the actual equations. It's a 2-compartment IV. And under parameters here, I just unclicked random effects because we're not going to fit any random effects here. We just have one profile for each species. And then you can convert to edit as textual. And you can see we've got a very nice start on the model. So I have to add a covariate for body weight. I have to change all these fixed effects and make them A, B, C, D, E. And then I have to put in the allometric equations. So rather than show me typing each of those, I'll show you what the final looks like. Maybe I can make that a bit bigger. So here's where I've added the covariate for the body weight. For the fixed effects, instead of having, again, volumes and clearances, I changed it to be the intercepts and slopes for the scaling models and put in the scaling functions. So it's a pretty straightforward way to do this. And then these values here, these initial estimates for the intercepts and slopes were the ones that came out of the Lin-Mix module that I just showed you a minute ago when we where we fit the logarithm of each of the parameters to the logarithm of body weight. Okay? And if you go ahead and run this model, here's the final estimates. And as we saw in the slide deck, we've got very, very good precision on all the parameters. The one with the highest precision is C, and it's at 23%. And what I did, and this will be in the model that we sent out, you've got the equations right here. So as you can see, C is the intercept for volume, D is the slope. So the intercept for volume had a 23% CV. Most of the other CVs are markedly smaller. So the fixed effects are estimated very nicely. One of the plots I always like to look at is the dependent variable versus the individual predicted values. That looks very nice. Here we're looking at observed and predicted overlaid for each of the species. And you can see that the fitted value is very, very good. And if we look at the QQ plot, we see that they fall very nicely on a line. So that's suggesting that the multiplicative error model that we used actually worked quite well. Okay, so again, it's pretty straightforward. We first fit a 2-compartment model to each species separately. We take those values and we fit regressions to them to get these initial estimates. And then we fit a combined model where we're able to combine across species by using allometric scaling to get the PK parameters for each species. And then lastly, I did a Dedrick plot. So in order to do the Dedrick plot, I need a body weight and dose. I wanted those on the same data set with the raw data. And then I did the data wizard. Now, the data wizard, this is exactly like we saw on the slides. For the Y variable, we normalized the body weight to the power of 1. In other words, for the simple Dedrick plot, we assume D is 1. And if you go to the second transformation, there's the scaling of the time variable. And again, we've got D minus B. D is the exponent for volume. B here is the exponent for clearance, but for the simple plot, we're assuming D is 1. And then we can just take those results and plot them. And that's our simple Dedrick plot. And then I took the same data and did a complex Dedrick plot. And when you do the complex Dedrick plot, you just allow D to be whatever the estimated value is. You don't assume it's 1. But the transformations are the same other than you're using the actual value of D. And we can get those data out. and plot those as well. And then as we saw in the slide deck, these values do overlay really quite nicely. All right, so that concludes the demo, but let's just recant what we did. We went through a mini lecture to talk about principles of allometry, but we also noted that you don't know in advance if allometry is going to work, although you may be in a situation where you're developing a sequence of analog compounds, and if you establish that allometry does work for one of the compounds, then it's quite likely that it's going to work for all the compounds in the sequence, assuming that they are structurally very similar. And then we went through a situation to explain how to actually do the allometric modeling, and we started with NCA to get some initial estimates for individual fits. We fit the individuals, each species separately, and then we just fit log-log regressions to them, and that gave us initial estimates then for the combined model. And the combined model, as we indicated, was, in this instance, was a standard two-compartment model, but volumes and clearances here are not the primary parameters. It's these intercepts and slopes, and then we use those to actually predict volume and clearance for each species. Okay, so that concludes the demonstration. So Bernd, I'll turn the podium back over to you for questions.
Okay, thanks, Anne, for this excellent lecture. It was lots of stuff, and I think this is was really highly educational. So thanks very much, Dan. And you know, the Q&A session is open, and so far we've got a few questions. There's one question then about the LinMix model. Could you go back to the first LinMix model and maybe explain once again the individual components and the regressor of the LinMix model, if you can share? Yeah.
Yes, I'd be happy to.
Yeah. Okay.
So in LinMix, essentially what you do, you have columns on your data set, and what you tell it then, which of these columns correspond to classification variables? That would be if you're doing like an analysis of variance. So if you had treatment, or if you had different conditions like period or sequence, those would be classification variables, and you would mark them here. And then if you are doing any type of covariate analysis, you would indicate which columns on your data set are regressors, and then also what your dependent variable is. So the only thing I told it to put in the model, there were no classification variables. The only thing I told it to put in the model was log of body weight, so it shows up here. So then what you can do is you just drag it down here. So now it's in the model. And it will default to have an intercept in the model, but if you wanted to do a regression without an intercept, you could simply click this box, okay? But otherwise, it has an intercept in it. And then you have the option of transforming the dependent variable for the regression. So here I'm telling it for each of these estimates, I don't want to just do the regression on them directly, but I want to do it on the logarithm of each of those. So I'm doing a log-log fit in order to get those initial estimates. Now, if I had other factors in the model, for example, if there had been classification variables, I could have said, well, I've got log of body weight, and then I could have clicked the plus sign and like drug treatment up here. So then I'm doing an analysis of covariance, where the covariate is body weight, and then the treatment would have been the treatment variable. But in this instance, we don't have any classification variables. So I'll pause there, Bernd.
All right, thanks, Dan. A few more questions. Maybe one is, I mean, you started to explain about this, but the question is, how would you implement this approach in pop PK direction? What would be the changes?
Right, so if this was a pop PK model, let me go back. Let's see if we can do this just quickly. So in this model, I'm going to tell it again, I have a 2-compartment model. Now it's population. I won't turn off the random effects, okay? But I will just go to textual directly. And then, actually, let me go back because I want the equations in there. So I want to unclick close form. So now we can edit as textual. So this is where then, you know, I could come here and I type in covariate body weight to put that in. And then here, let me go back and I'm just going to cheat here for a minute. I need these scaling values. So I can put those in, and then I have to modify all these parameters, all these, you know, to get rid of the volumes and make them A, B, D, E, et cetera. So I'll do one of those. So instead of volume, we'll do A first, and that would be a typical value of A, and you would have an exponent A to A, and then here I would have a fixed effect A. So that just put the A parameter in there, and I would just repeat that for each of these other fixed effects in the model. Okay. Then in your random effects, you would list all of these etas, so you'd have an eta for A, an eta for B, an eta for D, an eta for E, et cetera. And you'd put in the initial estimates here the same way that, you know, from fitting each species separately, we could use those as initial estimates. So I think that's, you know, I think that's pretty much it for how to go about it, unless there's a follow-up question, Bernd.
No, I think that's pretty clear. I mean, and if it's really required, we might provide an additional model text that we will publish on the Certara forum on the PopPK version of this. There's maybe room for just one additional question. If you have multiple subjects from each species, do you suggest to calculate the mean value first and then run the model, or would you suggest to run the model directly and fit individually?
Yeah, so if you have relatively rich data on individual subjects, and if you have an NLME license, I think the population analysis would be preferred, because then you can actually separate between and within subject variability, and that would be a better approach than fitting mean data if that data is available.
All right. All right. All right, I think we'll stop here with the Q&A session. Thanks very much, Dan. It was really very helpful, very educational. Before we close this session, just one reminder. We have another session. Our next session is planned for March the 2nd, where we're going to present on a turnover model. We will characterize turnover of an endogenous compound. So I will send out an invitation to all of you in the middle of next week, so you will have enough time to register for that event. And I think with this, I want to close this session. Thank you for your continued interest, and you may want to disconnect now. Thank you. Cheryl, can we go back into the practice session, please?